Okay, we're recording. All right. So the first piece that we talked about was operons, right? What are operons? Where are operons found? What type of organism? Right, prokaryotes, right? So operons are a system in prokaryotes. And the operon has four major parts to it, okay? First part is regulatory gene, right? Then the second part is a promoter, which is your start codon. Then we have the operator and the structural gene, right? All of this is DNA, right? So this is a sequence that's going to code for a protein, right? And in the case of the trip operon and the lac operon, they're coding for enzymes, okay? Different types of enzymes. So this regulatory gene, what does it code for? So this gene all the way over here, what does it code for the production of? The repressor, right? It's either going to be a lac repressor or a trip repressor, right? So if we're talking about the lac operon, this regulatory gene codes for this protein, okay? So this codes for the repressor, okay? And the repressor has a specific shape, right? And it also has a binding site. So what does that tell you about the repressor protein? What kind of protein is it? If it has two shapes, right? Allosteric, right? So this is an allosteric protein that is produced, in this case, it's produced as an active repressor. So if the repressor is active, what does that mean? What can it do? Right, it binds to the operator. So if a repressor protein is made in the active state, it has a structure that allows it to bind to the operator, okay? And when it's bound to the operator, this RNA polymerase can't get through, right? So what are we preventing from happening? What process? What?
Translation.
Not translation. Transcription, right? So this is preventing transcription because this polymerase normally wants to copy this gene, right, and produce the enzyme. So the way this is set up right now, this operon, is it on or off? It's off, right? So the lac operon naturally is in the off position all the time. So with this repressor in place here, this polymerase can't copy. So the operon is off, right? So the lac operon is considered what kind of operon since it's off all the time? Is it inducible or repressible? If it's off all the time. It's inducible, right? To induce means to start or to kickstart it to get going. So this is an inducible gene, which means it's off all the time. Now, what is going to trigger this to become activated? The presence of what substance allows this to become active? Say it again. Lactose, right? So if lactose now suddenly enters the system, okay, here's our lactose. It's going to bind to the lac repressor, right? The lac repressor has a binding site, right, that this lactose can attach to. When it does this, it changes the shape of the repressor. And the repressor no longer has the ability to bind to the operator, right? We pull that away. Now what can happen? Right, the RNA polymerase moves along here, and it copies these genes, right? What does it form? First, what is it copying this gene into? A sequence of what? What is it?
mRNA.
Yeah, mRNA, right? This is going to make mRNA, right? And then that mRNA gets translated into protein, right? We're going to form lactase, okay? So when lactose is present, right, the polymerase can read the DNA and transcribe it into messenger RNA, which then gets translated to lactase, right? Lactase then is going to increase in concentration, and it's going to start binding to the lactose and breaking it apart, right? This is going to digest all the lactose. So once that lactose is taken away, including this one, right? Now the repressor to reattach. And so what happens to the gene now? It's what? It's turned off again, right? Once this lactose is gone, now the repressor is activated again, okay? And now that polymerase, when it wants to come back here and make another copy, it can't, okay? And so we've turned it off again, okay? We turned it off because what substance was no longer present? Lactose, right. Because this consumed all the lactose, once the lactose is removed from the system, there's no need to make lactase anymore, right? We don't want to produce lactase because there's nothing for it to digest. So without lactose present, this turns it off, right? It binds back to the operator, and we stop making additional enzyme, okay? So this way, we only make the proper amount of enzyme that's needed to digest the lactose. Once all that lactose is gone, now we go back and suppress the protein, and it no longer works again, right? And it stays off until what happens? When does it get turned back on again?
When lactose is present.
Right, when lactose is present, right? So it's an inducible gene. It's off all the time, and it's only being activated whenever lactose is present. How do we feel about that? Is that better? Do you like my animation so far? Is it working? Does it work? Okay. So now let's play around with a different one. So now we've got a regulatory gene that's producing this thing, which is the TRP repressor. Now, what's unique about the TRP repressor protein compared to the LAC repressor protein? What's different about them?
Not allosteric.
It is allosteric,
Oh.
but it's not what? And if you could read this, good for you, but it's not. It's inactive, right? This protein is produced in an inactive form, which means it can't attach to the operator, okay? It's kind of a strange concept to think of that, okay, well, you have a protein, you're making a protein, but the protein doesn't work. We actually have a lot of proteins that are made this way, where they're formed in the inactive state, and then they're only activated when needed, right? So that means now that this repressor, right, when it comes over here, it doesn't stay, right? It'll come here, bind, and then quickly come off, because it doesn't have the ability to truly attach. Which means that even though this repressor is present and it's not attaching, what does that mean for RNA polymerase? Right, this is going to just sit here and constantly copy this, right? So in the TRP operon now, this gene is always actively transcribed. What we're doing is we're producing the genes that we're copying, produce the enzymes that make tryptophan, okay? So what the TRP operon is doing is producing enzymes in a pathway that makes the amino acid tryptophan, okay? Now, when the tryptophan levels start to go up, right, because we're constantly making tryptophan, right? Tryptophan is always being made because we're always transcribing this gene and translating it into the enzymes that make tryptophan. This tryptophan eventually makes its way to the repressor. What happens when it touches the repressor and binds to it?
Changes shape and binds to the operator.
Yeah, this changes shape, okay? And now it's activated, and this can attach to the operator with the TRP attached to it, right? And now what happens to the gene? Can't transcribe, right? So what happens now? It stops, right? And so now this gene is turned off, right? So now we've repressed it, okay? Now, the cell needs tryptophan to make proteins, right? This is an amino acid, so it's going to use the tryptophan. Once all that tryptophan is used up, right, including this tryptophan, because the cell needs tryptophan, right? Now we turn the gene back on, okay? This gets removed, right? And now we start copying the DNA and transcribing it into RNA, right? That RNA codes for these enzymes that make tryptophan, okay? And we're going to start producing tryptophan again, right? And the tryptophan levels are going to go up. As the tryptophan levels increase, right, what happens to this gene as the tryptophan levels go up?
Are you repressing it?
Yeah, you're going to repress it, right? Because you're going to get to a certain point where you're going to make enough tryptophan, right? You get to what's called a critical concentration. You make enough tryptophan that you can now shut down the gene by activating the repressor, okay? So in the presence of tryptophan, the repressor is active, okay? Whereas with the LAC operon, in the presence of lactose, what happens to the repressor? It stops working. It's inactive. So they're opposites of each other, right? In the TRP operon, when the end product, right, the protein that you're coding for, when the protein is made in the TRP operon, it inactivates the repressor. In the lac operon, when you make lactose, so when lactose is present, right, you're going to inactivate this, okay, so that you can copy the gene. Okay, so it's two different things, right, going on, because in the lac operon, this is produced in the active form, okay, your repressor is already active. In the trip operon, the default position of your repressor is inactive. It's produced as an inactive protein, which means this is always on in the trip operon, okay? And in the lac operon, because the repressor is activated, it's always produced in the active form, and so it always keeps the gene off, okay? Does that make sense? Are we good with that, right? So this is an example of negative feedback, right? So what's happening is the thing that you're producing in both cases shuts down or starts up the protein. It has an opposite effect on what you're doing. So for example, with the lac operon, it's off all the time, right? Now, lactose is present, right? You've turned the gene on with the presence of lactose, right? What does this gene do in response to the presence of lactose? What does it make? Lactase, right? You're going to make lactase. What does lactase do to lactose? Breaks it down. So if you break down lactose and get rid of it, what happens to your gene? It gets it what?
It's repressed again.
It's repressed again. You turn it off, right? So you're negatively affecting the gene's response because the gene's initial response to lactose is, okay, let's make lactase. Fine, you're making lactase, that's going to turn the gene off, right? That's negative. If it was positive feedback, what you would do is continuously make more lactase, right? You'd keep adding to it. Go ahead.
Wait, so like how is it considered?
So, say it again.
How is it considered inactive?
The repressor,
Oh, okay, so lactose itself is not considered.
right, and that's going to inactivate this. So once this is inactivated, now you can get the gene going. You good? Thanks. Okay. So it's negative feedback because if it was positive, the production of lactose, the production of lactase would eventually cause you to digest lactose and break it down. If it was positive, you would continue to make more lactase. Okay. The same thing with tryptophan. With the trip operon, you're making tryptophan, right? Once you get to a critical point, you cap it and you turn it off. Right? If it was positive feedback, that what would happen is the production of tryptophan would stimulate the gene more and make you produce even more tryptophan, right? Which would stimulate the gene more and make you make even more. But that doesn't happen, right? But that's what positive feedback would be. You would just continually build whatever the gene codes for, right? The gene would never turn off. And with the lac operon, the same thing would happen, right? You code for lactase. The more lactase you make, supposedly, the more lactase you make, the more lactose you break down. But if it was positive feedback, you keep making even more lactase. You'd never turn it off, right? But that's not the case. So that's what negative feedback means, is whatever you're producing in the end here is going to ultimately shut down the gene, okay, and cap it, all right? Do we get this now? Is this better? Like you can, and again, you're going to need to know what a promoter is, right? That's a start codon. So that's where the RNA polymerase attaches, okay? And again, the regulatory gene, that's responsible for making, there goes the repressor, okay? So the production of these repressor proteins happens by this gene. And in the trip operon, the trip repressor is always produced in the inactive form, okay? What activates the repressor is what substance? What activates this repressor? Tryptophan, right? So the production of tryptophan, when it gets to a higher concentration, activates this, okay? The lac repressor is active already. So what's going to inactivate it? Lactose, right? Lactose inactivates this repressor, and that's what allows you to turn the gene on. Tryptophan activates this repressor, and that's what allows you to turn the gene off, okay? So trip is always repressible, lac is inducible. Are we good? Does that work? Okay. The next piece, let's move this over. I'm just going to keep this up here because I can't keep moving things around. Is eukaryotes, right? Okay. What are the things that increase gene transcription? So in a eukaryotic cell, what factors are going to stimulate transcription of a gene or increase the chance that you're going to transcribe that gene into RNA? This is all like the PIDX1 stuff. What is it? Switches, right. Yep. Okay, so you've got activator switches, right? Okay. What do those activator switches connect to? What kind of protein? Because it's all proteins, basically, right? So you have your switch. What attaches to the switch?
Yeah.
Activators, right? Right, which is the activator protein, right? This is also known as the enhancer, right? Okay. What else do you have that increases the chances that you're going to transcribe? Yeah, mediators, right?
Oh, and gene transcription factors.
And transcription factors, right? Okay, transcription factors, they're going to bind to the start codon, right? And that's where you're going to be able to copy your DNA. Let me move this down a little bit so you guys can see, okay? So the transcription factors bind to the start codon, right? Activator proteins bind to the switches. What do the mediators do?
Bridge the gap between the activator and the general.
Yeah, yep, they connect the activator to the transcription factor, okay? So these mediators, they connect the activator to the transcription factors. I'm in the room. There's one more thing that you can add to the DNA, which increases transcription. And it's a compound that you add to the DNA, which actually will increase the chance that you're going to copy the DNA at a certain location.
Promoted leader?
You have the promoter there already, right? But then these are things that can bind to the area of the promoter to increase transcription. Starts with an A. Yeah, acetylation, right? I'm going to kind of write this over here, right? So acetylation, right? That's going to increase and enhance the chances that you're going to copy the DNA, okay? Acetyl groups allow these transcription factors to bind more efficiently. It increases the affinity of those transcription factors for those regions, okay? What's the one thing that's really going to decrease transcription? And it has to do with epigenetics. A lot of it was what we talked about in epigenetics.
EPA, for example. Or like harmful substances.
Okay, but what does that make the DNA do? Remember when you were had the whole licking the rats? What was changing on the DNA? Methylation, right? So if you methylate, right? Adding methyl groups, it causes the DNA to coil up, and that's going to reduce transcription, okay? Demethylating is going to increase transcription, all right? So we'll add that on this side too. Demethylation, right? Taking away those methyl groups through, whether it's through a chemical substance that actually binds to the methyl and pulls it away, or you're having something like an event like the nurturing of the rats, where it triggers enzymes to pull it away. When you demethylate, that's going to increase transcription. When you add methyl groups, okay, you're gonna reduce transcription. Demethylating is going to increase, okay? And so when you methylate, that causes DNA to condense, right? It coils up tighter, and it makes it tougher to copy the DNA. When you demethylate, that allows the DNA to relax a bit, takes out those coils, and you have more access to those genes, okay? Better with this now? Is there anything you guys are still confused on, or does this make sense now? I think doing the chart's better because now you can kind of see like, okay, these are the things that make things go up. This is what makes it go down, okay? So after transcription, you have really the siRNA and the miRNA, right? And the miRNA and the siRNA, I'll write that up. Actually, I'll erase this once you guys are done. Got it? You good? We good? Can I erase this part? Okay. So Just this little thing here. So post transcriptional regulation, right? That deals with RNAi. Okay, and there are two kinds of RNAi. There's the miRNA and the siRNA. Okay, both of them inhibit translation. Okay, and basically the difference is miRNA has the base pairing is not exact, whereas SI, it's exact base pairing. The siRNA sends a signal to degrade the mRNA, okay, where the microRNA, what this does, this blocks the ribosome. Okay, both of these, the job is to inhibit translation, right? So for both of these, the whole goal is to not make the protein anymore, stop producing the protein. And if you already transcribed, it's okay, let's cut the protein off before we're done making it. So you're only making part of a protein. So you're making an incomplete, non-functioning peptide fragment that really doesn't do anything, okay? And that's it. Block ribosome. So it's going to prevent the ribosome from moving any further, reading the messenger RNA to produce a protein, okay? And that's it.
So the siRNA binds.
Very specifically, yep.
And then the binding triggers the signal for the mRNA to be
Degraded, yep. What happens is these enzymes come in and they just start chopping it up. Yep, okay? And it's very specific. It's precise.
like outside enzyme.
Yeah. Yep. Yep. Okay. That's it. I'm trying to think if there's any other. Is there anything else anything that you guys feel still unsure about that you want to go over again? You're pretty solid with it? Okay. All right. All right, that's it. Cool. Okay, I'm going to pause the video.